सभागार में उपस्थित आप सभी सम्मानित जनों का हमारे इस चल रहे 3 दिवसीय राष्ट्रीय संगोष्ठी में हार्दिक स्वागत एवं अभिनंदन है। संबंध स्वास्थ्य विज्ञान संकाय There is information for excuse me मैम फार्मेसी कहाँ भेजना है? फार्मेसी के बच्चे रूम नंबर 46 में जाएंगे। जितने भी फार्मेसी के बच्चे यहाँ पर उपस्थित हैं रूम नंबर 46 के लिए जाएंगे और जो सीटें वैकेंट हो रही हैं पीछे से आप सभी उसको कवर कर लेंगे, बीच में सीटें वैकेंट नहीं रहेंगी। संबंध स्वास्थ्य विज्ञान संकाय एवं फार्मेसी संकाय के संयुक्त तत्वावधान एवं ट्रांसलेशन बायोमेडिकल रिसर्च सोसाइटी लखनऊ के विशेष सहयोग द्वारा संचालित 3 दिवसीय राष्ट्रीय संगोष्ठी के द्वितीय दिवस के अवसर पर आगामी तकनीकी सत्रों की श्रृंखला में हम यहाँ चतुर्थ तकनीकी सत्र में उपस्थित है, जिसमें आज हमारे साथ अध्यक्ष स्वरूप प्रोफेसर के रामचंद्र रेड्डी जी व उनके साथ सह अध्यक्ष के रूप में डॉक्टर दिनेश कुमार जी का आज मार्गदर्शन प्राप्त होगा। सर मैं आप दोनों से आग्रह करूंगा कि कृपया करके आप मंच पर आकर अपना स्थान ग्रहण करें। प्रोफेसर के रामचंद्र रेड्डी सर एंड डॉक्टर दिनेश कुमार सर। ठीक है, आज हमारे बीच सह-अध्यक्ष के रूप में डॉक्टर दिनेश कुमार सर, एसोसिएट प्रोफेसर सेंटर ऑफ बायोमेडिकल रिसर्च, प्रेसिडेंट ट्रांसलेशनल बायोमेडिकल रिसर्च सोसाइटी के ही ये हमारे इस बायोनेचर 2023 के को कन्वेनर भी हैं। मैं डॉक्टर शशिकांत सर से आग्रह करता हूं कि वो मंच पर आएं और सर को श्री राम जन्मभूमि के मोमेंटो से सम्मानित करें। मैम आप चलिए, वही नहीं है। ठीक है, हम लोग के बीच अध्यक्ष स्वरूप रेड्डी सर कुछ कारणों से अभी विलंब है, इसलिए मैं कार्यक्रम को आगे बढ़ाने हेतु मैं आज के व्याख्यान के लिए जो आए हैं हमारे प्रोफेसर शरद कुमार मिश्रा सर, जिसे मैं आग्रह करूंगा कृपया वो मंच पर आए। सर इसकी
ओके काम करेंगे अनुपा, आप इसे बोलो ना फोटो ले।
मैं व्याख्यान शुरू होने से पूर्व आज के जो हमारे
ये चीज वर्किंग है
सर वो रिकॉर्ड वक्ता है उनका मैं आप लोग के समक्ष उनका परिचय करूंगा। प्रोफेसर शरद कुमार मिश्रा सर, renowned academic from former head department of biotechnology, दीनदयाल उपाध्याय, गोरखपुर यूनिवर्सिटी, PhD in study of produces inhibitor from legumes, department of biotechnology, दीनदयाल उपाध्याय, गोरखपुर यूनिवर्सिटी in 2006 and MPhil from Jawaharlal Nehru University in 2000. And I
think we have already gone through my introduction.
Okay, sir.
In the last session.
Yes, sir, yes.
So thank you so much for inviting me. Okay, so we should start.
Please, please. Okay, thank you. So since morning you have been listening lectures. We had 2 lectures in the first session, and I hope that you have got a lot of things. Wonderful lectures. I will talk about neem, Azadirachta indica. How many people know about neem? And those who don't know, आपको नीम नहीं पता है? कोई ऐसा भी है जिसको नीम के बारे में नहीं पता है? कितने लोगों ने नीम नहीं यूज किया है? सब लोगों ने किया है? चलिए, बहुत बढ़िया है. So last lecture we were talking about how we can utilize the modern scientific technologies to validate our ancient wisdom or ancient knowledge about the treatment. So neem, this is basically a household tree. It was. It's one of the very important plant, and the history, if you go for to the history of this tree, it is quoted around 5000 BC. And the leaf extract of this plant, they were also reported from Mohenjo-daro. So if you see how this plant has been quoted, how important this plant is. Though we all see neem plants most of the places, but we are mostly unaware of the benefits of this plant. So Ayurveda, it says Sarva Rog Nivarani, the universal healer or curer of all ailments, most of the diseases, almost all diseases. Arishtha, this is the perfect, complete and imperishable. Nimba, Nimbati, which means to give good health. This seed, this dark ticket, neem was so important in our history that a dark ticket was, issued in the name of a tree in the Mughal period. So you can imagine. This neem is also known as village pharmacy because this is a tree of unbelievable wonders. And we can also call it as biochemical factory. The single tree is a kind of biochemical factory that contains several thousands of chemical constituents that are attributable to the miracles. So it is a miracle tree, neem. The Unani scholars, they knew about this plant and they called Shajare Munarak or the blessed tree. Persian scholars, they called it Azad Dirakht Hind, the noble or free tree of India. Now, the neem has been declared the tree of 21st century by United Nations. And US National Academy of Science also recognizes neem's medicinal value, and in 1992 reports neem a tree for solving global problems. So just one plant of our country can be used to solve a lot of problems, health problems. This is just a simple slide which showing the effect of neem. It can be anti-cancerous activity. It has role in anti-cancer activity, anti-diabetic activity. Sorry. Anti-diabetic activity, neuroprotective activity, anti-inflammatory, antiviral, antibacterial, antifungal, antioxidant, antimalarial, hepatoprotective, whatever you can imagine of. In the morning, we have seen neuroprotection, neurodegeneration. Then we have seen liver, hepatic problems. All these things, they can be, protected by using neem. Now, all the parts of the neem plant, they can be used. They have medicinal value, medicinal properties. This is a list of leaf, bark, flower, fruit, twig, gum, seed pulp, oil, and all those things. And these are the medicinal uses of this plant, neem plant. If you use leaf of neem, then it can cure leprosy, it can solve eye problems, intestinal worms, anorexia, biliousness, skin ulcer, epistaxis, epistaxis. So all these problems can be solved if you only chew the leaf of neem. नीम का सिर्फ पत्ता आप खाइए, गांव में लोग खाते हैं, ये सारी प्रॉब्लम्स आपकी दूर हो जाएंगी, आपको किसी डॉक्टर के पास जाने की जरूरत नहीं पड़ेगी। क्योंकि जो मॉडर्न मेडिसिन है उसके दो प्रॉब्लम्स हैं, उसका साइड इफेक्ट भी बहुत ज्यादा है, लेकिन यदि आप नेचुरल नीम का लीफ लेते हैं, उसको यूज करते हैं, तो दिस कैन बी यूज्ड एज ए मेडिसिन। तो हम सभी जानते हैं, हम सबको अवेलेबल भी है, कॉस्ट हो शायद, बट वी आर नॉट यूजिंग इट। बार्क ऑफ लीफ, इट इज एनाल्जेसिक, यदि आपको थकान हो, थकान दूर हो सकती है। वर्म्स कुड कंट्रोल फीवर्स, ट्रीट लॉस ऑफ एपेटाइट, भूख नहीं लग रही है आपको। क्लीन टीथ, balance the cough constitution, treat malaria, skin diseases. All these diseases, they can be cured if you use the bark of neem. Flower of neem, this is the only part which is having a better taste, while all the other parts they are bitter in taste. The flower of leaf, it is antioxidant property it has, treatment of belching, nausea, anorexia, intestinal worms. The Ayurveda says that his flowers are magical for the eyes and beneficial for treating headaches and skin diseases, bile suppression and phlegm. This is the use of bark of neem. If you go to the fruit, you must have seen the fruit of neem plant. It relieves from the piles, intestinal worms, urinary disorder, epistaxis, phlegm, eye problem, diabetes, leprosy, wounds. Twig. पहले हमारे जो लोग थे, they used to use neem as a toothbrush. We forgot. Now we are using toothpaste and toothbrush, but this is the natural toothbrush which protect us from cough, asthma, piles, phantom tumor, in which there is water filled in the lung, spermatoria, obstinate urinary disorder, and diabetes, just by using the twig of the neem. What we did, we shifted from our traditional lifestyle to some modern lifestyle. We are using so many toothbrush and toothpastes, while we have a natural toothbrush with us available everywhere. There is no need of toothbrush, no toothpaste. 7 days experiments with herbal cleaning twig, datun, जिसको बोलते हैं, which can help to prevent tooth decay and gum diseases. So if you use this datun, your smile is beautiful because your teeth are healthy, shining. So we can use this. See, this is the benefits of chewing neem twigs. क्या benefit है? It whitens the teeth, antimicrobial and tongue cleaner. If you use the twig of neem, all these things can happen. What happens actually? The corners of the neem twig chewed first. जिसने use किया होगा उसको पता होगा. Then during the process, the chewed corners work like the bristle of the brush and they clean the corners of the teeth. Chew करने से क्या फायदा है? When we are chewing, it releases the medicinal agents present in the neem twigs. And neem twig comes in contact with your tongue, contains antibacterial agents that protect us from the harmful microbes in mouth. So it is just not a toothbrush or datum, it is also a medicine if you use the twig of neem. Gum of neem, it protects us from ringworm, scabies, ulcer, wound, and skin diseases. Neem oil that is available. And what is the function of neem oil? We use the neem oil to treat worms, insects, mosquito bites. The neem oil acts as an insect repellent. जो हम आजकल mosquito, Gorakhpur is known for mosquitoes also, other than. So we can use these neem products for repelling the mosquito. Its antifungal properties can help us to get rid of hair disorders. Hair की बहुत problem है. Dandruff and treat head lice. It also used to treat urine infection, urine disorders, as well as stomach and intestinal worms. And many other products can be prepared as an ingredient by using the neem products. तो यदि आपको अपने बाल बचाने हैं, तो you have to prevent hair fall, so you have to use neem oil. Dandruff-free hair, cures skin diseases, hair and skin, promotes hair growth, promotes anti-aging, powerful antifungal. All these are the properties of neem oil. सबसे important जो है, जो आपके काम का है, वो ये है anti-aging effect of neem. If you want to delay your aging, आप जल्दी बूढ़े नहीं होना चाहते. कौन होना चाहता है? कोई चाहता है? Nobody. सब लोग चाहते हैं ना, मतलब ज्यादा समय तक हम हमारे पास एनर्जी हो, and I should be young. कोई नहीं चाहता कि फेस पे रिंकल्स हो, एजिंग साइंस आए. पीपल आर. तो ये नीम का जो इफेक्ट है, द नीम लीव्स full of antioxidant, moisturizing triglycerides and vitamin E, which makes them a perfect anti-aging treatment. If you want to reduce the aging process, you must use neem. They reduce the appearance of wrinkles, जो चेहरे पे wrinkles होते हैं, fine lines and dark spots, and makes your skin look firm and toned. So that is the property in neem. Other than other remedies, neem can reduce your aging process. Besides boosting collagen, collagen, जो हमारे फेस पे जो wrinkles आते हैं because of the collagen. Collagen is a protein. So there is change in the collagen, and that gives rise to wrinkles. So boosting collagen and improving skin elasticity, they also protect skin from aging signs caused by sun exposure and UV radiation. तो आजकल बहुत सारे sun से protect करने के लिए बहुत सारे creams आते हैं, but we can use neem for that. Regular use of neem leaves paste can slow down your aging clock and rejuvenate skin. So you can go home and prepare a neem face pack with neem paste and sandalwood powder and use it twice a week to achieve a maintained youthful and radiant skin. So neem oil prevents premature aging. So just imagine that how important it is. और ये नीम कब से यूज हो रहा है? सदियों से यूज हो रहा है हमारे देश में. लेकिन if you go back, if you read 90s में US, यूरोप में नीम को पेटेंट कराया जाने लगा. नीम के बहुत सारे जो प्रॉपर्टीज हैं, they started getting patent. Then we got up, Indian people, they came to know that neem is being patented, the properties of the neem is being patented, antifungal property of neem that was patented in Europe. So the people of India, they opposed it, they filed suit. In some of the cases, the patent was retracted. Some cases, we didn't get success. This is our traditional knowledge. The people of Europe and US, they are getting patented. So if you want to use, we have to pay them. So hamara wisdom. We never sold our wisdom. India never sold. If you go through the contribution of India in science, ancient time, whether it is Shunya, discovery of zero or decimal or binary system or anything, lot of big list. Even the aeroplanes, the electric battery, all these things, they were given by Indian people to the world. But we never sold it. We never asked for money. But now the time has come. If you create any intellectual, matter or material, you will get money. So that's why nobody thought of. The people outside the developed countries, they started for patenting. So we have to be careful. This is our tree. We have been using. Nobody should take away from us. नीम जूस शायद ही कोई पीता होगा, ठीक है, लेकिन if you drink neem juice, can reorganize and reboot one's digestive process. It can enhance one metabolism, efficient in breaking down body fats, enhance body excretion system, purifies blood by removing unwanted bacterial growth because of antibacterial and antiseptic enzymes that are present in the neem. Because of all these, the neem juice is considered as one of the most beneficial juices to consume. Finally, what is neem?
Connected.
Neem is the best kept beauty secret of ancient India. Neem is the best kept beauty secret of ancient India. The neem leaf and oil contains a synergy of soothing and nourishing essential fatty acids, omega, and cooling and astringent antioxidant. आपके पूरा health, the neem only can take care of your health. So we have a tradition. We use tulsi. In older time, every household used to have tulsi, used to have neem, all these plants. But now the science is, validating that why we should use tulsi. Why should we use neem? It is not on the Vishwas. It all has scientific basis. Now, looking at the properties of the neem, we thought of, let us see the role of neem, if it can play a role in dengue, protection of dengue virus. So we had a report, a small report published, in which we came through that the neem extract, leaf extract, it has anti-dengue activity. So we thought, ठीक है, neem extract anti-dengue activity है, लेकिन उसमें कौन से molecules हैं, which molecules are there, which actually show anti-dengue activity. तो डेंगू की बीमारी अभी आई थी, आपको पता होगा, बहुत ज्यादा, in fact in Gorakhpur also, the dengue is spread like anything, and we don't have any medicine for dengue till today. We just use some other medicines, symptomatic medicines for the treatment of dengue. So now we started this work, identification of antiviral compound from Azadirachta indica against dengue virus. NH3 protease. डेंगू के बारे में आपको पता है, it belongs to the Flaviviridae family, and it is the causative agent of dengue fever. And there are 5 virus serotypes which are available, and they cause hemorrhagic fever and dengue shock syndrome, and they are transmitted to individuals by nibble of Aedes aegypti female mosquito. So mosquito act as a carrier for this transporter. The dengue has also emerged an increasing fitness problem in the world for which no specialized drug is available in the market. This is the mosquito which is the culprit. So I have shown this so that you can identify it and keep yourself away from this mosquito to avoid dengue. So this is the mosquito. Now, if you talk about the dengue virus, it has single-stranded RNA genome, which is packaged and surrounded by 180 copies of the 2 glycoproteins. There are 2 types of the proteins which are present in this. There are 10 proteins. Dengue virus is made up of 10 proteins. 3 are structural proteins: capsid, membrane, precursor, and envelope protein. And 7, they are non-structural proteins: NS1, NS2A, NS2B, NS3, NS4A, NS4B, and NS5. And these proteins, non-structural proteins, they coordinate the assembly of new virus once the virus gets inside the host. So these are the structural proteins. These are the proteins which are helpful in the assembly of the viral particle. This is the diagrammatic representation, M protein and capsid and this. These are the proteins, non-structural proteins. Now we focus on non-structural protein NS3. NS3 is a protein which is present in the dengue. And if somehow we are able to block this protein, inhibit this protein, the dengue virus particle will not be able to multiply. So we focused on NS3, and it has been already recognized as a potential therapeutic target for the discovery and development of novel antiviral agents against dengue infections, because it is important and required for virus replication. So if you are able to inhibit NS3 protein which is present in dengue, we will be able to block the multiplication of this virus. So we have to search that molecule. The molecule which can go and bind to NS3 protein, protease, and block it, inhibit it, and inhibit the replication of virus. The NS3 molecule of dengue, that is, consists of 2 domains. One is protease, the other one is helicase. But we are, concerned with the protease, NS3 protease, which is responsible for the replication of the virus. Now this NS3 protease domain, it depends on a cofactor that is known as NS2B. This one, NS2B, for its optimal catalytic activity. So there is a complex structure that is called as NS2B, NS3 protease. So it exists as a complex, and we have to search inhibitors for this complex, NS2B, NS3 protease.
Connected.
So you see this diagram, helicase domain. This is the helicase domain. This one is the protease domain. And there is a red ribbon-like structure. If you see, this is NS2B cofactor. So NS3 protease as well as NS2B cofactor, they exist as complex. The functioning of this NS3 protease depends on NS2B cofactor. So now we are going to target the dengue NS2B, NS3 protease. We need to search for novel inhibitors from different plants. We can search different sources. So in area of antiviral drug discovery, there are various reports on antiviral activity of various medicinal plants against dengue virus, and that has been done. Now, neem plant is one of the among those medicinal plants which has been reported to show potential antiviral activity against dengue. But the extract is showing anti-dengue activity, but we do not know the active compound, actually the compound in the neem which is responsible for inhibiting the dengue virus replication. So that is what we have done. And if you want to search a molecule, bioactive molecule from any plant extract, so there is one approach. First, you can go for in silico study. By using computational methods, you can identify the molecules which are interacting, like in this case NH3 and NH2B complex interacting with any active molecule of the neem. So we can go first in silico and then we can go for in vitro study. So preliminary work when we started, that was published in vector-borne disease. And we identified 5 triterpenoids of neem plant. Nimbene, disacetylnimbene, disacetylsalanine, azadirachtin, and salanine. These 5 triterpenoids were checked for their in silico inhibitory potential against NH2B, NH3 protease by using molecular docking approaches. And on that basis, the result we obtained that nimbene, disacetylnimbene, and disacetylsalanine, they were found to be, having inhibitory potential against NH2B, NH3 protease complex. So out of the molecules which we screened, only 3 we got: nimbene, disacetylnimbene, and disacetylsalanine, which shows the inhibition. So now we moved ahead, and for in silico study, there is requirement of collection of structural data, receptor and phytochemicals of neem from various available databases, PubChem, or there are so many databases from which you can find out the molecules, chemical structure of the molecules which are present in neem. They're all stored there. Now the computational screening of inhibitors from neem phytochemicals against NH3 protease, the molecular dynamic simulation, and then in vitro evaluation of selected inhibitors for this. So now we published one review paper, Genomics, Proteomics, and Evolution of Dengue Virus in Briefings in Functional Genomics. During our work, now for in silico study, we have to collect the data, and the data, protein data bank, that is a popular data bank for 3D structure of proteins, and PDB, that was we used to collect the 3D structure of NH3 protease. So we were searching the inhibitor of NH3 protease enzyme from neem. So we need a molecule in the neem which can go and bind to NH3 protease of dengue. So we need first, for in silico study, we need the structure of NH3 protease. So for that we went for protein data bank, PDB. Then the second step was to collect the ligands which can bind to NH3 protease by using PubChem database, and most of the compounds which are found in the neem, they are present in this PubChem database, and we obtained those structures from this. Now, after getting both the structures, NH3 protease as well as the compounds from the neem plant, we went for molecular docking experiments by using several softwares. molecular dynamic simulation. And then we, after doing this molecular in silico study, we went for in vitro antiviral assay. And for that, we used BHK21 cell line to see the effect of molecules from the neem on dengue virus. So before we go for using this as a drug, we first see what is if there is any cytotoxic effect of these molecules on the cell, because we cannot use as a drug if it is toxic to the cell itself. So for that, we used cytotoxic activity of these molecules, camphorol 3O beta rutinoside and epicatechin and quercetin. They were determined by MTTSA. Now, detection of intracellular virus by immunofluorescence that we performed. So now, if we see the in silico study, the structure contains, if we talk about NS2B, NS3 protease, which was retrieved from protein data banks, this structure contains A and B chains. A chain is NS2B and B chain is NS3. If you see the diagram, this is the structure of NS2B, NS3 protease. It shows two chains, A and B. A is the red color, which represents NS2B, while purple color is representing the NS3 protease domain. Now, the active site forming catalytic triad residues are also there. That is serine 135, the histidine 51, and aspartate 75. So that is the catalytic triad which is present in this protein. Now, we collected around 40 phytochemical compounds, 3D structure of 40 phytochemical compounds from Azadirachta indica neem for analysis. These are the structures which we obtained from the data bank, 40 chemical compounds. So now if you see that, in summary, the NH2B, NH3 protease is there, which we collected from the PDB, 3D structure of this NH3, NH2B protease, and we targeting the Azadirachta indica, any molecule if we have in Azadirachta indica, which can be used for identify, which can bind to the NH2B, NH3 protease and inhibit this protease by molecular docking and molecular dynamic simulation. And these were the molecules which were used: camphorol, rutin, hyperside, epicatechin, and so on. So all these molecules were used for binding with the NH3 protease. So if they bind, they will inhibit this protease and viral replication will not occur. Now, when we found the binding energy calculated, the camphorol 0.9, 0.5, rutin 0.9, hyperoside 0.7. So this is the list of the molecules and their binding energy. How strongly they bind with the NH3 protease, that gives the idea about that. So when we found this result, you see the earlier finding which I showed you, nimbine, diacetylnimbine and diacetylsalanine, their binding energy was this much. So when we went for second round, then we found much better result, and that provided the evidence that the ligand molecules from the neem, they have a strong binding affinity and higher molecular stability to the target protein, and therefore these compounds can be evaluated for in vitro for antiviral activity. So this is simply the interaction, in silico interaction, how this is the molecule which is interacting with the several amino acid residues of the NH3, NH2B protease. So how this is interacting and binding. This is for routine. This is for hyperoside, epicatechin, quercetin. So these are the interactions in silico which we did. Now in the above findings, these molecules were used for their interaction with the NH3 protease, camphorol 3-ortho-beta-rutinoside, routine, hyperoside, epicatechin, quercetin, glucoside, quercetin, camphorol, glucoside, maris, all these compounds from the neem, they were used to for their binding affinity with the NH3 protease, and they, from as a direct indica, they showed good molecular interaction with NH2B, NH3 protease. Now, this strong hydrogen bonding between these compounds, NH3 protease, were detected with one of the residue of catalytic triad I showed you in the figure, histidine 51, aspartate, and serine 135. That makes the catalytic triad to which these ligands can go and bind, can interact. Some other conserved residues like phenylalanine and tyrosine, arsenine, asparagine, this 152, and glycine 153, they also found to play important role in the substrate binding. Some other new additional also they were reported. So now we focused 3 compounds of different binding energies range were selected for in vitro validation and antiviral activity against dengue. तो जो हाईएस्ट बाइंडिंग एफिनिटी जिन मॉलिक्यूल्स की थी उनको हमने फिर सेलेक्ट किया इन विट्रो स्टडी के लिए, and these were camphorol 3 or 3-O-beta-rutinoside, epicatechin, and quercetin. These were the binding energies of these molecules. Cytotoxicity assay जब हमने किया तो how we did, we used basically BHK21 cell line. BHK21 cell line used as a model to study the effect of these molecules, camphorol, rutinoside, epicatechin, and quercetin on the dengue virus. Now these cells were pre-treated with the non-toxic concentration of these compounds, like quercetin 1 to 10 micromolar of the quercetin, 10 to 100 micromolar of camphorol, rutinoside, and 0.1 to 1 millimolar of epicatechin. So cytotoxicity, if you see, the camphorol didn't show any cytotoxicity. They were the virus, the molecules, they were incubated for 24 hours as well as 48 hours. If you see the range from this 0.01 micromole to 100 micromole. Epicatechin, it also didn't show that much cytotoxicity, but in the case of quercetin, we found that it shows the cytotoxicity at the concentration 100 micromolar concentration. So BHK20 cells, 21 cells were there. They were cultured with the different concentration of camphorol, epicatechin, and quercetin for 24 and 48 hours. And the cell viability was assayed, and it was found that these 2 molecules, they didn't show any cytotoxicity. Only quercetin showed the cytotoxicity. The quercetin exhibited 100% toxicity at 100 micromolar concentration. On the basis of that, we decided to study the concentration, these concentration for further study. Camphorol 10 to 100 micromolar, epicatechin 0.1 to 1 millimolar, and quercetin 1 to 10 micromolar. Now for study of antiviral assay, we used immunofluorescence assay for this, and BHK21 cells, it was first infected with the dengue virus, 5 MOI, and they were, after the, they were also incubated with the drug. So first we treated the cells with the drug or molecule which we selected in our study, and then we infected this with the dengue virus, and we observed significant inhibition of dengue infectivity upon treatment with the lower concentration of these compounds, with greater reduction in dengue infection evident at higher doses of each compound. So जब हमने इन molecules को use किया anti dengue activity के लिए, we found that it inhibits the infection. Now if you see here, quercetin that inhibited, if you use 1 micromolar concentration, 60% inhibition of the infection of dengue virus. When we use 10 micromolar concentration of quercetin, more than 75%, inhibition of infection. The cells get protected. Camphorol, if we see 10 micromolar, more than, above 50% inhibition of infection. And when we use 100, more than 77% inhibition of infection by the dengue virus. And here in epicatechin, when we use 0.1 millimolar, we got little less than 50%, while when we use 1 millimolar, we got around 66% inhibition of the infection of dengue virus. And this was the fluorescence result in which we used the cell line, BHK21 cell line, infected with the dengue virus. And then we used dengue virus monoclonal antibodies for the illumination. So in fluorescence, we can see this. Now we come to like camphorol, rutinoside, and quercetin. They showed more than 50% inhibition in dengue infectivity at both concentration, while epicatechin showed less than 50%. Overall, these drugs showed reduced infectivity of dengue virus. Precisely, if you see 100 micromolar camphorol, rutinoside 77.7%, 1 millimolar epicatechin 66.2%, and 10 micromole quercetin 75%. So now we have identified these 3 molecules which have the potential, which can be developed as a drug, which can be used as a lead molecule for the development of drug against dengue virus. So identification of potential inhibitors against dengue virus, NH2, NH3, NH2B, NH3 protease, a prime therapeutic target for anti-dengue virus discovery. It is an urgent requirement. And in the present study, 12 potential inhibitors from Aedes recta indica were predicted by molecular docking and MD simulation experiments. And the antiviral activity of 3 compounds, camphorol 3-O-beta rutinoside, epicatechin, and quercetin, they were performed using in vitro experiments. So at this point of time, we can say that these above compounds have a very good binding affinity and higher molecular stability with the dengue, NH2B, and NH3 protease, and also possess wonderful in vitro anti-dengue activity. Hence, camphorol 3-O-beta rutinoside, epicatechin, and quercetin can be used for the development of powerful medications to combat the deadly infection of dengue virus. So this was also published in Journal of Biomolecular Structure and Dynamics, anti-dengue infectivity evaluation of bifluoronides from Aedes recta indica. So this work was performed by the culture the fluorescence microscopy was done by with the help of Professor Luzi from Hyderabad, and this work was done by Vivek, my student, who is well known about the bioinformatics. And so
आपकी वक्तव्य से और भी ज्यादा समझ आई कि किस प्रकार के अब सभागार में यदि किसी को भी प्रश्न मत करना हो तो एक एक कर जी सर।
Thanks to Professor Misraji for elaborating historical aspects of neem, which has been mentioned in Sanskrit language as Nimba, and he has given various parts of the neem plant, their therapeutic uses, since long how it has been used in various systems like Ayurveda and Unani. Later on, he also explained docking study or in silico study how the components has been isolated and their study over the dengue virus has been elaborated with the proof of the publications in international journals. As in terms of Professor Misraji, neem is the very important drug, and neem has played a very important history in central government of India. As he told, I just want to brief regarding the neem history in the central government. Neem and Haridra, that is turmeric, has been patented by other countries. In response to that, Indian scientists quoted that neem has been used traditionally since ancient days. But the United States of America has asked that where is the proof? In the published media, published form. So, in that condition, generate the proof of evidences of all traditional medicines, not only the neem and other medicines of Ayurveda and Unani and yoga and Siddha systems. The CSIR has given the responsibility by the Parliament. The issue was raised in Parliament and matter was discussed elaborately. The project was to the CSIR called TKDL, Traditional Knowledge of Digital Library. Under the traditional knowledge of digital library, all the therapeutic usages mentioned in traditional knowledges like Ayurveda and others has been recorded in 7 international languages like English, German, French, etc. The reason is that the United States of America, in the case of neem, has mentioned that where is the literary proof, published proof for the neem in acceptable language like international language like English and publications with the evidences. In response to that, this project has been mentioned, given to the CSIR and TKDL was formed. I was worked in TKDL as a task force member for 10 years. And during that time, round about 150 patents of different scientists has been cancelled, including Colgate company, which has been used Colgate one variety of toothpaste by using jaipur, that is nutmeg. So now the present scenario is that TKDL, traditional knowledge of this digital library, has displayed many number of Ayurvedic drugs, formulations, their therapeutic uses in internet, and one scientist who is working, they have to study that one, they have to take their literary study in that, then only the for new work, any patenting, if they want to go, new work which is not mentioned in our tradition of Ayurveda can be taken out. This session by Professor Misraji is very interesting, highlighting the ancient science of Ayurveda into the with evidence by doing the in silico and docking study. I am very much thankful to the Professor Misraji for elaborating all the scientific information, including the historical evidences of neem, and also many of the students who has got opportunity to hear this session. they have got benefited, including myself. I am from Ayurveda faculty from Banaras Hindu University. So very, very thanks. And now session is opened for discussion. If anybody rise to any question, if they want to discuss any matter pertaining to the need, now Professor Misraji is ready to answer. Thank you.
Take my mic. Thank you, sir, for a very informative lecture. So my query is, apart from the beneficial impacts of NEEM, many papers has reports that NEEM has also induces germ cell apoptosis, and NEEM has also used as a contraceptive. So how you can justify the only beneficial impact of the NEEM? So can you explain it, sir?
Yeah, in fact, I have shown this benefit also. For family planning purpose, we can use neem also. And one of the scientists, if you know him, Professor G.P. Talwar, he was going to make a contraceptive vaccine. So he also tried neem for contraception, and so we can use this demerit of neem for our own benefit, for family planning and so on. And moreover, we need to study more for this. That is the property in the neem, like which part of neem and what is the molecule which is responsible for this, reduction in the size, the number of sperms in the male. So that can be looked into, and but for contraception it can be used.
So now I request the student audience, please participate in the discussion. So in this presentation, sir has covered almost computational as well as computational bioinformatics and in vitro experiment. So because we have time, 20 minutes before lunch, because we in the second speaker is not here. So I request students, can you ask the questions, or I may ask? Okay, please. Please give the mic. What is your name?
Good afternoon, sir. Myself, Srivastava from Biomedical. Sir, बात अपनी loss of hepatitis पे कैसे help करता है?
लॉस ऑफ हेपेटाइट जो होता है, आपको भूख लगती है, कभी कभी आपको भूख लगती है तो आप खाना खाते हैं, कभी कभी भूख नहीं लगती है लोगों को, अक्सर ये शिकायत होती है भूख नहीं लग रही, तो उसको बढ़ाने का काम करता है, हेपेटाइट को बढ़ाने का काम करता है, इट एक्ट्स एज एपेटाइजर। ठीक है, तो यदि आपकी भूख खत्म हो गई है तो उसको यदि आप नीम का लीफ या बार्क लेते हैं तो उससे आपकी हेपेटाइट बढ़ेगी। समझ में आई बात?
Thank you, sir.
Anybody else?
Any other student?
Okay.
आगे जाइए, आगे।
Good afternoon, sir. My name is Shaggy, and my question is, why do we use only neem for chickenpox, and how does it work on it?
Why do we use
Only neem at ancient time.
अच्छा, वो ट्रेडिशनल,
Yes.
because neem has that property to protect us from chicken pox. At that time we didn't know, right?
And sir, what is the process that?
But see that people, अभी तो क्या है, वैक्सीन है, ड्रग्स हैं, तमाम चीजें हैं, उस समय तो कुछ नहीं था, तो उस समय के लोग जानते थे कि नीम को यूज करेंगे तो शायद उससे प्रोटेक्शन होगा, उससे बचेंगे। ठीक वैसे ही जैसे तुलसी है ना, आप अपने घर में लगाते हैं, तो तुलसी बहुत सारा एंटीबायोटिक होता है, एंटीबायोटिक प्रॉपर्टीज हैं तुलसी में, इसलिए घर में लगाते हैं, उसकी पूजा करते हैं। पीपल का वृक्ष लगाते हैं, उसको काटते नहीं है, बोला जाता है कि नहीं काटना है, तो उसका भी साइंटिफिक बेसिस है। वैसे ही नीम है, आप विलेजेस में जाइए, इसको विलेज फार्मेसी बोलते हैं, हर घर के सामने नीम या उसके पीछे नीम हर गांव में पहले होता था, अभी कम हो रहा है, तो उसके पीछे रीजन है। People used to use the twig of the neem as a datum, and they used to use several ways. जो leaves गिरती थी उनको फिर जलाते थे, फायर एक तरह से वो ऑर्गेनिक फ्यूम या स्मोक जनरेट होता था और जो इन्वायरमेंट था वो ठीक होता था। All these things were used traditionally, but now we need to focus, we need to validate scientifically. That is what we are trying to do.
सर बट जो वैक्सीन लगती है बोलते हैं कि मतलब वो बीमारी के बाद, बट चिकन पॉक्स की जो वैक्सीन होती है फिर भी चिकन पॉक्स दोबारा हो,
जो वैक्सीन होती है दोबारा हो जाता है। वैक्सीन देखो, वैक्सीन क्या है, अभी एंसेफेलाइटिस के बारे में जानते हो आप, होता है कि गोरखपुर का इंसेफेलाइटिस, तो इसका वैक्सीन है, वैक्सीनेशन कराया गया, फिर भी हो गया इंसेफेलाइटिस। अब वैक्सीन उसको कोल्ड चेन एक होती है, इसमें कोल्ड चेन में मेंटेन करना है, वो कोल्ड चेन टूट गया तो वैक्सीन क्या हो गया, इनएक्टिव हो गया। So that can be one reason, especially if you talk about the viruses. These viruses, they keep on changing. They are very notorious. If you develop a drug, if you develop a vaccine, they change themselves so that the vaccine or drug is no more effective against them, especially the retroviruses. So we have to track the vaccine or the drug that is developed against which virus, like कब develop किया गया, क्योंकि 5 to 10 years में वो वायरस कंप्लीटली चेंज हो गया, तो आपका वैक्सीन काम करेगा नहीं करेगा। ठीक है, जो रेजिस्टेंस आपने पढ़ा होगा ना, रेजिस्टेंस डेवलप हो जाता है या मॉडिफिकेशन हो जाता है, so that is the reason sometimes it doesn't work. ठीक है, थैंक यू।
हेलो सर, आई एम प्रशांत कुमार फ्रॉम बीआरडी। सर, क्या नीम को हम लोग कंटिन्यूअसली कितने दिन तक यूज कर सकते हैं, ताकि उसका कोई साइड इफेक्ट ना हो?
जिंदगी भर यूज करिए, बस डोज में। एनीथिंग above its dose is toxin, कोई भी चीज हो, आज ड्रग के रूप में काम कर रही है, उसके लिए आप एक्स्ट्रा लोगे तो क्या हो जाएगा, आपके लिए पॉइजन हो जाएगा। तो एक फिक्स्ड डोज में आप लाइफ लॉन्ग यू हैव टू यूज इट।
कुछ योगी होते हैं, उनका कहना है कि इसको 15 दिन कंटिन्यूअसली यूज करने के बाद 15 दिन तक छोड़ देना चाहिए।
हो सकता है, आई एम नॉट अवेयर ऑफ दैट। हो सकता है, बिल्कुल।
इसके ऊपर कुछ रिसर्च नहीं है?
इस पे तो कोई मेरे जानकारी में कोई रिसर्च नहीं हुई कि 15 दिन यूज करो 15 दिन छोड़ दो, बट वी कैन सी इट। ठीक है, आई एम नॉट अवेयर ऑफ दैट फैक्ट।
और किसी का प्रश्न?
अभी सर एक्सप्लेन कर रहे थे कि वायरस वैक्सीन काम नहीं करता, ठीक है, तो majority of these drug designing studies where we develop antiviral, okay, also these natural compounds or antiviral drugs, they also do not able to kill the virus, okay, just we are having antiviral drugs to keep the viral replication or virus life cycle in control, so that our own body immunity take over and do not allow the virus to go or to propagate further. Okay, so this just keep in mind when we are developing anti-bacterial drugs. The aim is different. When we are developing antiviral drugs, our aim is objective is different. We are developing some inhibitors so that we can target the non-structural proteins. Okay, structural proteins we cannot develop targets because they are not useful. targeting the structure proteins mostly. They are used for vaccine development. So in case of viruses, when you have to develop the vaccines, you target the surface proteins or the structural proteins basically. When you have to develop this inhibitors, you go for non-structural proteins. Okay. So I'm asking one question so that we can get involved in the discussion. So any student in the audience can help me with the definition of a non-structural protein. Any student who is confident that he can tell the definition of non-structural protein. Now I think all will wake up. What are non-structural proteins? Sir, showed one slide. No? Dengue virus has 3 structural proteins and some 6, 7, 8 non-structural proteins. So what is the difference between structural and non-structural proteins? Any student who had listen to the talk, please help me with the definition. Simple one-word definition, one sentence definition. This is the sir. This is the non-structural protein. No. So how many of you want to go for developing drugs for diseases or antiviral drugs? Anyone who is interested to go in this area of drug designing, please raise your hand. Okay, 3 are fine. Please get up. We will discuss. This is how we will enjoy the science. Okay, please get up. Okay, so can we have that slide? Slide virus slide. Simple proteins which are expressed by this viral genome within the host cell. These are categorized in the non-structural. Okay, non-structural doesn't mean those are not functional. Those are fully functional proteins. But we call them non-structural proteins only because they are not the part of that viral particle present on the surface or on our when we छींक से है ना बाहर आता है तो पार्टिकल होता है उसमें वो absent रहता है उसमें नहीं होता वो जो वायरल पार्टिकल का स्ट्रक्चर है उसमें जो प्रोटीन्स होते हैं उनको हम स्ट्रक्चरल प्रोटीन्स बोलते हैं ठीक है अब ये सबको क्लियर है कि वायरस में स्ट्रक्चरल प्रोटीन और हम नॉन स्ट्रक्चरल प्रोटीन कहां से आ गए ठीक है तो नॉन स्ट्रक्चरल प्रोटीन्स है जब वायरस किसी होस्ट सेल लंग सेल को इन्फेक्ट कर सकती है हिपेटोसाइट कोई कई वायरस होते हैं बहुत स्पेसिफिक होते हैं हिपेटोसाइट्स को ही इन्फेक्ट करेंगे तो कोई एक एग्जांपल बताएगा कोई बता दो सिंपल सा एग्जांपल है हेपेटाइटिस ए वायरस हेपेटाइटिस बी वायरस हेपेटाइटिस सी होता है ना वो सिर्फ जाके ज्यादातर उसको नुकसान ही सिर्फ उसमें जाके हेपेटिक सेल्स को ही जाके इन्फेक्ट करेगा क्योंकि उसका तो सरफेस का जो मॉर्फोल हुआ है जो उसके बाहर प्रोटीन्स होते हैं मैच करते हैं उससे रिसेप्टर से तो अंदर एंट्री में फैसिलिटेट करते हैं ठीक है तो स्ट्रक्चरल नॉन स्ट्रक्चरल प्रोटीन में डिफरेंस तो समझ आ गया ठीक है अब सर ने बहुत अच्छा काम किया कि नीम का जो प्लांट था उसके सर ने एक्सट्रैक्ट नहीं सर ने क्या किया जो ऑलरेडी अवेलेबल इंफॉर्मेशन थी इनके जो लैब के मेंबर्स थे उन्होंने आइडेंटिफाई किए कितने नीम के प्लांट में कितने कंपाउंड्स नेचुरल कंपाउंड्स हो सकते हैं भांति भांति ठीक है तो दे इन हाउस लाइब्रेरी ऑफ 20 to 30 compounds or 30 compounds okay and one thing you all students have to keep in mind the structures available on the PubChem the 3D structures which sir showed that those all are solved in organic solvents when they have to crystallize chemists use some or these scientists who extract these they use different solvent conditions to crystallize these compounds and they publish they get the structure in crystal form and then they submit the data but those structures may or may not be have the same conformation in our biological system Okay, when they get into the cell, may in the lung cell or body cell. Okay, so and one step which I think I should tell you, that there is comes geometry optimization. Whenever you are involved in drug designing projects, whenever you are taking any structure from the PubChem, step follow the geometry optimization step. Okay, and it will tell you this protein not this molecule, basically this natural compound has 10 conformations, possible conformations. Okay, then you can use some other tools to see in biological system where mostly there is water, so which conformation of that ligand or that drug molecule is most possible. Okay, or alternatively you can have all the 10 conformation in your screening protocol. Okay, so that will keep your study more reliable, so you are not missing any conformation which could be there in the biological system, but the crystal structure is different from that existing in vivo conformation. Okay, that point please keep in mind. Okay, now coming to here, sir have mentioned this NS1, NS2. These are non-structural proteins. Okay, so now as we know, these non-structural proteins, they are expressed in the host cell. Means डेंगू वायरस कहाँ अटैक करता है, कौन सी सेल को अफेक्ट करता है, कोई है आइडिया? कहाँ? लीवर को? सर सर मुझे खुद नहीं मालूम है. I worked on influenza viruses, so they mostly involve the lung cells. Okay, so dengue virus is blood cells or
blood cells. अच्छा he's क्या 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 कहना क्या है?
डेंगू वायरस किन सेल्स को टारगेट करता है, कौन सी सेल्स?
डेंगू वायरस बेसिकली ब्लड में इन्फेक्ट करता है, ठीक है, और जैसे all the viruses they are specific, they have a ligand and they have specific receptors on the different types of cells. जैसे कोरोना वायरस, कोरोना it infected which body part? Lung. Why? Because there was a receptor on the lung tissue against for the coronavirus. So that is how the specific viruses, like encephalitis virus is here in Gorakhpur, where it infects brain, because brain has the receptor for this virus. So that is how the different viruses, they have a different receptors, and they infect through those receptors. They make entry into the cell.
Thank you, sir. So these proteins are expressed by viral genome in the host cell. Okay, inside the host cell. Now these proteins perform their own functions, and then they use the host machinery to produce various parts of that viral particle, and then those parts get assembled, and then the cell gets bursted, and these particles get released. Okay, this is the viral cycle. Okay, and in the study, sir has used an approach to stop that path. That some one protein functioning, if we can able to stop, we can stop this cycle. Okay, and this is how our drugs, antiviral drugs work. We use some antiviral drugs to stop the functioning of these non-structural proteins, so that the virus do not get replicated at its own speed, and by the meantime our own immune system starts developing antibodies so that to eradicate the virus from our system. Okay, so I think we have now reached at the close to the lunch, so we can now thank sir for his excellent presentation. And
Thank you. One minute. Thank you, the organizers, for inviting me for this lecture. Dr. Sunil, who very kindly invited me, Dr. Nupma Ojha, and Dr. Dhananjay, Dr. Dinesh. So I am very much thankful to the whole organization team for giving me this opportunity to interact the legendary personalities. I can see sir from Mumbai. So, and dear students, and I give you a message that please use neem further. How many people will use now? So neem use neem for your benefit, health benefit, and make other people also aware. So thanks a lot. Thank you so much.
मैं सर महोदय अध्यक्ष महोदय जी से अध्यक्ष महोदय जी से आग्रह करूंगा कि वो प्रोफेसर शरद कुमार मिश्रा जी को विश्वविद्यालय के मोमेंटो और सर्टिफिकेट देकर सम्मानित।
थैंक यू सर, बड़ा अच्छा लगा सर आपसे मिलकर। ये भी थैंक यू सर, थैंक यू सो मच, बहुत अच्छा लगा सर।
धन्यवाद सर। कुछ कारणों से हमारे प्रोफेसर रामचंद्र रेड्डी सर इस कार्यक्रम में जब हुए जिसके कारण उनका परिचय नहीं हुआ है। मैं आप सबके समक्ष उनका परिचय रखूंगा। प्रोफेसर रामचंद्र रेड्डी सर रसायन वैशल्य कल्पना के प्रोफेसर हैं, संस्कृत के महाज्ञाता है एवं 24 महान आयुर्वेदिक पुस्तकों के लेखक हैं। डॉक्टर रेड्डी ने आयुर्वेद को वैज्ञानिक ढंग से अंतर्राष्ट्रीय स्तर पर स्थापित करने का अथक प्रयास किया है। अब मैं फार्मेसी डिपार्टमेंट के प्रिंसिपल डॉक्टर सिद्धार्थ सर से आग्रह करूंगा कि वह आज के इस चौथे डिजिटल सेशन का समापन नहीं होता है और इस इंफॉर्मेशन के लिए